This is a production of Cornell University.
Thanks very much, Teresa, and I really want to thank both you and Gillian for the invitation to visit Cornell virtually. I've been to your campus several times and really enjoyed it, and I'm also an alumnus of the Cornell Medical School in New York City, so I have an affinity for the university. And I don't know if you can see it, but I have my Cornell hat right here, and we can put it on. I think that might be appropriate for the seminar today. And I have my fleece, which is from the Weill Cornell Medicine as well. So we are doing our best to advertise here in North Carolina, the excellent educational institution that you all are associated with. Let me go ahead and share my screen. And does that show the first slide of the PowerPoint? Excellent. So as Teresa mentioned, I want to share with you today a story about our work on a novel form of drug resistance, antimicrobial drug resistance, that we discovered in a basal fungus, Mucor circinoloides. And let's see. I want to begin the story with Max Delbruck. And many of you know that Max was a physicist who transitioned to work in genetics and molecular biology. He was part of the phage school of molecular biology. This is a pamphlet from a centennial celebration of his birth that was held in Spain a number of years ago that I had the opportunity to attend. And Max is famous for many different contributions to science, but one of those is illustrated here. This is a paper published in Genetics in 1943, and many people refer to this as the fluctuation test. So Salvador Luria and Max Delbruck were interested in what is the basis of mutations that occur in biological systems. And they distilled this down to the simplest experimental system. They used E. coli as the bacterial host, and they studied mutations that were occurring that conferred resistance to a lytic phage, T1. So you grow cultures of the bacteria, you expose them to the phage, and then plate out. And if you take one subculture and test it multiple times, you see a little bit of experimental variation in the number of resistant colonies that arise. But the key experiment was to grow multiple subcultures, expose those to the phage, and then you see a much more dramatic variation in the level of resistance. Particularly, some of these have many, many resistant isolates, and these are a jackpot effect. And it's attributable to when the mutation arises in the growth of the culture. If it arises early, you have many resistant mutants. If the mutations arise late, you have very few or no resistant mutants. So this was the experimental setup. There was a lot of detailed statistical analysis, but it led them to conclude that mutations spontaneously occur in populations, and then selection acts upon those to select the resistant ones that are better suited to survival under the stressful conditions. They went on to share the Nobel Prize with Al Hershey in 1969 for discoveries concerning DNA replication and genetic structure of viruses. And a key aspect of this was their discovery of the basis for mutations occurring to phage resistance and generalizing that to thinking about mutations in all populations for all organisms. Al Hershey, you'll remember from the Hershey-Chase experiment conducted with Martha Chase, in which they showed that the DNA of phages was the genetic material rather than the protein. So after Max Delbruck won the Nobel Prize, he shifted entirely his research focus and program to work on the organism that's shown here. This is called Phycomyces blakesleanis, and it's a fungus. It's a basal fungus. It used to be part of the Zygomycete phyla of the fungal kingdom. Now we know it as the Mucorales. And he was particularly interested in studying this as a model for sensory perception because these long sporangiophores are very responsive to environmental cues. And this includes light, touch, wind, gravity, and even the presence of adjacent objects. And this is showing the light responsiveness of the organism. This isolate on the left is a blind mutant, and the one on the right is responding to a white light source off the edge of the plate. And the sporangiophores reorient their growth towards the light source. So when Alex Idnerm was a postdoc in my lab, he was studying light sensing and other properties in Cryptococcus, but his weekend hobby was to come in and work on Phycomyces. He just fell in love with this organism and contributed a series of publications on Phycomyces. The first two were in collaboration with Luis Corrochano and Arturo Eslava's groups in Spain, and involved identifying the genes and the proteins involved in light sensing. These are mutants that have been isolated in Max Delbruck's lab in the 60s, MAD-A and MAD-B, and they encode orthologs of the white collar 1, white collar 2 protein complex that senses light in Neurospora. Alex went on to identify the mating type locus in this fungal species. It had been earlier named the sex locus. These were studies published in Nature. We'll return to the sex locus as part of the main story in just a few minutes. And then finally, there was a large consortium that came together that was organized by Luis Corrochano and Victor Gar and Igor Grigoriev, who sequenced the genomes of both Phycomyces and Mucor succinyloides. This was published in Current Biology just a few years ago. And what it revealed is that these species have undergone repeated whole genome duplication events. We know about the whole genome duplication event that punctuated the evolution of Saccharomyces cerevisiae, that's critical in producing duplicated genes and paralogs with many interesting biological functions. These species have undergone 2 whole genome duplication events, and that turns out to be a recurrent theme throughout biology and other species as well. And one of the consequences of that is it's dramatically expanded the repertoire of signal transduction proteins that are available in these species, as well as some other pathways, including the RNAi machinery, as we'll see in a few minutes. So I want to turn our attention then to think about another species that lies within the basal fungi. This is the fungal tree of life here. We know a lot about the Ascomycota and the Basidiomycota because of model species that populate those 2 phyla, things like Saccharomyces cerevisiae, Ustalagomatus, Candida albicans, and Neurospora, for example. These have a monophyletic origin from this green point here, and these are referred to as the dicaria. They descend from a last common ancestor. But all of these other sort of blue and orangish balloons here are other phyla that we know about now from molecular phylogeny in the fungal kingdom. And we're going to be talking about the Mucorales, indicated by the red arrow here. And both Phycomyces and Mucor succinyloides and a human pathogen, Rhizopus oryzae, lie within this group of fungi. And these are understudied organisms, and the application of whole genome science has really opened a unique window to be able to study them in detail. So we pivoted when Su-Chan Lee arrived in the lab as a postdoc from working on Phycomyces to focus on Mucor, because it's dimorphic, it's a human pathogen, and you can transform it and disrupt genes by homologous recombination. And Phycomyces is strictly hyphal. It's not a human pathogen, and so far no one's been able to transform it. So it remains a challenging model system. So Zygomycete fungi as a class cause devastating human infections. Some of those are illustrated here. And part of the reason these are so devastating is because we have a very limited armamentarium of antifungal drugs, really limited to just amphotericin B, posaconazole, and isobuconazole. In some cases, you require surgery to control these infections. And there's a broad panoply of organisms that cause Zygomycetus infections, and they're diverse and understudied. We chose an area in which we wanted to focus in exploring the biology and the pathogenesis of Mucor succinyloides. And we are studying calcineurin. It's a conserved calcium calmodulin activated protein phosphatase that in previous studies had been shown to be required for pathogenesis in other fungi. In Mucor, this petri dish here shows the standard hyphal growth mode, and under the microscope you can see the branching hyphae that are typically formed. But when we grew the organism on media containing a calcineurin inhibitor, the natural product FK506, you see this small restricted yeast colony. And when you look under the microscope, these are multi-budded yeast cells. So this calcineurin inhibitor completely blocks the dimorphic transition from yeast to hyphae. It turns out that this had been discovered earlier, in fact, by Louis Pasteur in 1876, rediscovered in 1962 by Solomon Bartnicki Garcia, namely that if you grow Mucor under anaerobic conditions with a little bit of CO2, sorry, these slides keep changing. If you grow it with anaerobic conditions, you also see a multi-budded yeast. So the calcineurin inhibitors completely bypass this normal anaerobic condition that's required to lock the organism into the yeast phase. So armed with this observation, Cecilia Schertz, as a rotating graduate student, set out to understand how this might operate. So we know a lot about FK506 as a natural product. It diffuses into the cell, it binds to a protein called fkbP12 to form a high affinity complex, which then binds to and inhibits the calcineurin phosphatase, a complex of a catalytic or regulatory subunit and calmodulin. Similarly, the other natural product, cyclosporin, binds to cyclophilin and also inhibits calcineurin. These are natural products that have come to prominence because they're used as immunosuppressive drugs in transplant recipients. They also have extremely potent antifungal activity, and we've been focusing on that in our studies. We know a lot about the structures of these complexes. So on the right and the left, these are the actual X-ray crystal structures determined by other groups, and more recently for the fungal orthologs by our group and collaborators. Here's fkbP12 bound to FK506, docked on to a common surface of calcineurin. It binds to a region that's distinct from the active site, and it makes these inhibitors exquisitely specific for inhibiting this phosphatase and no others. And this is the mechanism of both antifungal activity and immunosuppressive activity in the human immune system. So Cecilia set out to isolate resistant mutants to this growth inhibitor FK506. So again, without drug, you see a hyphal growth mode. In the presence of the drug, you see a yeast colony. And if you wait, you see drug-resistant hyphal sectors emerging. So we purified these drug-resistant sectors and characterized them. As you might imagine, some of them result from standard Mendelian mutations in the known drug targets, and some isolates of this category are shown here. This is a deletion of fkbP12. These are point mutations or other substitutions on either calcineurin B or calcineurin A that confer resistance to FK506. Those allow us to essentially map out the surface that the drug inhibitor complex is docking onto on calcineurin A and B. We can model how these mutations confer resistance. So this confirms that this drug is acting through the known targets in this fungal species, but it doesn't really shed a great deal of new light or information. It's interesting, but what turned out to be even more interesting was a second class of drug-resistant variants. And one such isolate is shown here. This is the control medium. Here, the wild type is sensitive to FK506. This isolate is resistant to FK506, and it's also cross-resistant to a second drug called rapamycin. And like FK506, rapamycin binds to fkbP12 and inhibits a target in the cell, in this case called TOR. So the fact that these isolates are resistant to both drugs implicates fkBP12 in their, the basis for their resistance. So about 70% of the drug-resistant isolates have Mendelian mutations in the 3 known drug targets, but about 30% did not. And the question is, what are these unusual drug-resistant isolates? By Western blot, using an antibody raised against yeast fkBP12 that cross-reacts with the Mucor protein, we could show that fkBP12 is missing in these drug-resistant isolates. And by Northern blot, we could show that the gene is not being expressed. So the implication that fkBP12 is somehow involved is borne out by these findings. At this point, Cecilia came to my office with a troubling look on her face and said, you know, I'm really frustrated by these mutants because when I passage them, they keep reverting to being drug sensitive. And she was worried and upset about this. And I said, I think that might be interesting. I think we should look more carefully at this. So this table summarizes what we found. 2 wild type strains, no matter how long you passage them, they always grow as a yeast in the presence of FK506. They're sensitive. If we delete the fkBP12 gene, it's a rock stable Mendelian mutant. It always is resistant to FK506. And 3 of her epimutant variants, you can see with some stochasticity for which passage they revert from being resistant to being sensitive again. So these isolates, unlike a Mendelian mutation, are unstable and transient. And the images at the bottom here show they become sensitive again to FK506, and they also become just as sensitive as the wild type to rapamycin. Now, interestingly, when these epimutants revert to become drug sensitive, the expression of fkBP12 is restored at both the protein level and at the mRNA level. So they can become drug resistant, they shut off expression of the key drug target, and then when they revert, magically it comes back on. So we considered a number of different possible models for unstable drug resistance, and I want to focus on the one that turned out to be the explanation. We knew that Mucor had an active RNAi pathway from work going on in Spain at the University of Murcia in the labs of Victor Gar, Rosa Ruiz Vasquez, and Santiago Torres Martinez. They and their colleagues had been identifying components of the RNAi pathway, including dicer argonauts and RNA-dependent RNA polymerases, contributing a series of publications on this topic. And so we entertained the hypothesis that these drug-resistant isolates might involve spontaneous RNA silencing of the fkBP12 gene. To test that, we made small RNA from the wild type, the various forms of the epimutant grown in FK506 in the resistant form or in the reverted sensitive form, ran them out and probed them with a probe specific for antisense RNA to the fkBP12 gene, encoded by the fkbA gene. And remarkably, you'll see that all of these epimutant isolates have abundant small RNAs to the fkBP12 locus, but when they revert to be sensitive, those all disappear. We made small RNA libraries and sequenced them so we could look at the repertoire of small RNAs across the entire genome. And what this lower panel shows those displayed across the fkBP12 gene, which has 3 exons. The wild type has no small RNAs to the gene. The 3 different resistant epimutants all have abundant small RNAs to both the sense and antisense strand. And when they revert, those small RNAs go away. And notably, you'll see that the introns of the gene are not decorated by small RNAs, only the exons. And so it implicates the mature mRNA from the fkBP12 gene as being the target for the small RNAs. Now, importantly, we did not see any spreading of small RNAs to adjacent genes. We didn't see amplification of small RNAs in the drug-resistant isolates compared to the wild type or the sensitive isolates anywhere else in the genome. There certainly are other small RNAs, many of which decorate transposons throughout the genome, but they don't change in response to the isolate becoming resistant and sensitive. So to a first approximation, the only thing that has happened is this gene has been silenced by RNAi. We went on to take a genetic approach. Sylvia Kahlo, who had been a student with Rosa and Santiago working on the RNAi pathways, came to the lab, brought all our mutants with her, and we proceeded to march our way through the RNAi pathways to look at which components are required for this epimutation process. And it was a straightforward experiment. We would isolate resistant isolates in the wild type or in an RNAi mutant, and then simply determine the percent that were due to epimutants. In the wild type, it's routinely about 30%, and in the RNAi mutants that are required for the pathway, it's below the limit of detection. So we could show that dicer 1 and 2, one of the RNA-dependent RNA polymerases, Argonaute-1, the QIP exonuclease, and the RNAi helicase were all required for the formation of these drug-resistant epimutants. We're interested in all of these, but I want to draw special attention to this helicase. It turns out that that gene lies immediately adjacent to the mating type or sex locus in Mucor, Succinyloides, and other related species. The open reading frame is not strictly part of the sexually dimorphic region of the mating type locus, but regions of the promoter, we think, differ between the plus and the minus mating type. So that's particularly intriguing that an RNAi component is linked to the mating type locus, given previous studies in Cryptococcus and other fungi that RNAi pathways are often activated during sexual reproduction. Now, this helicase turns out to be interesting in another respect, and that is it's the ortholog of a helicase previously studied in Neurospora that's been found to be required for an RNAi pathway that mediates meiotic silencing of unpaired DNA. And what's interesting is RNAi is the direct reciprocal blast hit to a helicase from Pombe, which itself is the best reciprocal blast hit to SAD3. But when we do this reciprocal blast hits between Neurospora and Mucor, it's the best hit in one direction, but it's the third best hit in the other direction. We think it's the bona fide ortholog of SAD3, but it suggests that sometimes you have to do more complicated phylogenetic analysis to ascertain this. And the domain structures of the helicases are shown at the bottom here. So we're interested in further studies on whether the sexual cycle of the organism will be linked to the process of RNAi-dependent epimutation. We could go on to show that some RNAi components are fully dispensable for forming epimutants. That includes 2 of the Argonautes, Argonaute-2 and Argonaute-3. They're fully dispensable, whereas Argonaute-1 is required. And then there's a third class of RNAi components that's perhaps the most intriguing. And in these mutants, there are 3 such mutants, 2 RNA-dependent RNA polymerases and an alternative dicer called R3b2. You find a dramatic increase in RNAi-dependent epimutants, and they're dramatically stabilized. And in fact, some of these have never been observed to revert. So instead of finding 30% epimutants, we find in the range of 80% epimutants, and they're stabilized. So we can take all of this information and marshal it into a model that depicts multiple different RNAi-dependent pathways in Mucor that function in parallel. We've been focusing on the epimutation pathway, but in parallel, Victor Gar and his colleagues have defined an alternative RNA degradation pathway. And notably, 3 of the unique components in this pathway are the same ones that, when they're mutated, result in channeling more events through the epimutational pathway. So we think these 2 pathways are competing for aberrant RNA to either degrade it or to channel it into the epimutation pathway. And you can fine-tune this by mutating these components, or we think possibly by regulating the function or the expression of these components in response to different environmental signals. Now, you may be wondering, how did this epimutant form in the first place? And we considered the same question. And in looking at the annotation of the genome, what's quite striking is right next to the fkbP12 gene is an adjacent gene, and they're in an inverted orientation. And in fact, the 3' ends of the messages overlap by 92 base pairs. And this provides the potential for this region to form double-stranded RNA. And so we thought, aha, this must be the trigger for dicer. Maybe the poly A tails are trimmed, maybe another enzyme extends these, and then you dice this, and then that leads to RNAi silencing. So to test this model, we deleted this adjacent gene by transformation and homologous recombination with the deletion allele shown here. And to our surprise, complete deletion of this region had no impact on the formation of epimutations at fkbP12. So a good model, but we think that we can exclude this model. And we went on to discover that there's a naturally occurring antisense message to the fkbP12 locus. That antisense, like the sense message, is destroyed when these epimutants form. When they revert, the sense and the antisense message return. And by generating cDNAs from this antisense, we can see that it's generated from the mature spliced fkbP12 mRNA. We think that an RNA-dependent RNA polymerase might be involved in generating it, but we've tested each of the single mutants that we have available, and they all still make this antisense. So I think it may require some biochemical approach of making enzyme and seeing which of these might be able to copy the other strand to generate this trigger for the RNAi pathway. So to summarize to this point, we've seen spontaneous FK506 rapamycin-resistant mutants can result from either Mendelian mutations that are stable or unstable epimutations. In the epimutants, there's no change in the fkbP12 gene encoding fkbP12. The mRNA and the protein are absent. There's abundant small RNAs. And we think that this pathway could be either regulated in some fashion, or it might be completely stochastic in the same way that Lurie and Delbruck envisioned that mutations that occur in a population are just stochastic and then selection acts upon them. And we think that it might be an antisense that serves as a trigger. In some rare cells or nuclei, the sense and the antisense come together. That's a substrate for dicer, and then an epimutant arises in parallel with Mendelian mutations arising from polymerase errors, for example. Now, we went on to generalize this in two ways, and the first was to show that this also occurs in a second species of Mucor. And this was of interest, but it was also a key challenge put to us by the reviewers and the editors of our paper that was ultimately published in Nature. This took us something like 7 months to accomplish, and this experiment came together over the very last part of that time frame. So it's quite amazing how being required to demonstrate something can serve as an impetus to do experiments that you might not have done otherwise. We showed in two closely related species. They were originally considered forma, but they've now been recognized as distinct species. Mucor succinyloides lusitanicus, now Mucor lusitanicus, and Mucor succinyloides itself, that we find epimutants at 30% in the species we started with, and an even higher frequency in the MCC species shown here. Now, very recently, Sybren de Hoog and Kirsten Voigt and Gret Walter have published this key paper in Persunia, where they've defined the species complex here for Mucor succinyloides. And we've been talking about succinyloides and lusitanicus, but you can see by the PS designation, which means phylogenetic species, there's 16 different species in this complex, of which at least 5 or 6 are human pathogens. So we're quite interested now. We've set up a collaboration with Grit and her lab to bring in isolates across the species complex and test the whole panel for formation of epimutations. Now, the other major advance in this paper is for many years you could do sexual crosses in Mucor, but no one had ever seen the spores germinate. So you could not do a complete sexual cross. And this is the first report of zygospore germination from some of the isolates reported in their phylogenetic tree. So we're also quite excited about that because for the first time we can do a cross and see if that might change the frequency of epimutations, and it might link it to that helicase that's right next to the mating type locus. I should mention that we've tested a lot of other stress conditions, and with the exception of infection, which I'll tell you about in a few minutes, a panoply of other stress conditions in vitro didn't seem to stimulate the formation of epimutations. My graduate student, Zanetta Chang, who was an MD PhD student, then extended this in another direction, and that was to identify epimutations in 2 additional genes. This relied on using a drug, 5-fluoroerotic acid, 5-FOA, that was popularized by the yeast genetics community because it selects for loss of function in the URA3 or the URA5 genes. And in Mucor, these are referred to as PURF and PURG. We spent quite a bit of time looking for 5-FOA resistant epimutants unsuccessfully. And then Zanetta took a page from the delineation of the RNAi pathways, and she started with those RNAi mutants that have a higher frequency of epimutants, a kind of genetic helping hand. So in the RDRP1 and RDRP3 mutant backgrounds, she was nicely able to isolate epimutants. This is showing the small RNA blots, either in the PURF gene or in the PURG gene. Upon passage, they revert to being drug sensitive and the small RNAs go away. And when she sequenced the small RNAs, they again decorate the sense and the antisense strands of the PURF gene or of the PURG gene. The PURF gene has only an exon, no introns. The PURG gene has 2 introns. And again, you can see the small RNAs are specific to the exons and not the introns of the genes. So that generalizes this now across at least 3 other loci in the genome total. And we think that epimutations might, in principle, be possible to form at any locus throughout the genome if you had a selection for it. Now, Zanetta went on to do 2 other experiments looking at epimutations in the mouse. And the goal of the first set of experiments was to take a resistant epimutant, introduce it into mice, recover colony-forming units from different tissues, and just ask, is the epimutant stable during infection of a mouse? And you'll see in liver, spleen, kidney, lung, there it's very, very stable. Virtually all her CFU are still resistant when they come out of the mouse. But the isolates from the brain, about half of them, have reverted to be sensitive. So there's something unique about infection of the central nervous system. The second experiment she did was to take a drug-sensitive isolate, infect mice, recover from the same tissues, and ask, is there any induction of drug resistance in these isolates coming out of different mouse tissues? And there was very little effect from liver, spleen, kidney, or lung, but from the brain you see about 7% of the isolates coming out of the brain form FK506 resistant isolates on a Petri dish. So there's something unique about the central nervous system that we think is inducing plasticity of the RNAi system, either for reversion or for induction. And it might have to do with, for example, expression levels of the competing RNAi pathways. At least we think that is a workable hypothesis to pursue. The last experiment that Zanetta did was to infect mice, recover colony-forming units from the brain and the liver, culture these for 4 days, 6 days, or 8 days in vitro, and then ask, are there any small RNAs that are induced by infection in the mouse? And from this heat map, you can see there's a series of 7 genes that have abundant small RNAs induced against them in isolates recovered from the brain. And it's for me, that was quite remarkable that this worked, because this is 4 days and 6 days and 8 days of culture after recovery from the animal. It turns out 6 of those genes are completely unknown, but one of them is an ortholog or a homolog of a stress-activated kinase involved in stress responses in other fungi. Work by Carlos and Maribel, who were in Victor Gar's lab and are arriving soon in Durham to join the lab as postdocs. They've been looking at RNA-seq and small RNAs in the context of macrophage infections. And what's quite remarkable is this BEP-A gene. You can see that its expression is reduced in macrophages. And we think that that could be as a consequence of these small RNAs coming on. So these would be naturally occurring epimutations against different loci induced in the brain in response to infection. Now, this is preliminary data. So this was done with Mucor lusitanicus in macrophages. This was done with Mucor succinyloides in the whole animal model. So clearly we have some additional work to do here of exposing both of these environments to isolates from the same species so we can do a more direct comparison. Our plans for this part then, and you can see that this is aim 2 of the R01 that's been submitted on this, is to study the epimutations in the context of the brain, both in macrophage cell culture models, in blood-brain barrier models, in a brain organoid model with our colleague Debbie Silver, who's located 1 floor below our lab at growing brain organoids. And then finally to microdissect different regions of the brain rather than treating the brain just as one whole tissue, to ask whether or not there are specific regions of the brain in which these epimutants are reverting or being induced. We're also planning to delete these 7 novel genes and test whether or not there's an impact on the infectious cycle by generating a Mendelian mutation at those loci that are being impacted by an RNAi epimutation. So I want to summarize that epimutations are general and occur in 2 related species. They've been found so far in 3 genes. They can be, like mutations, spontaneous and stochastic. And they can also be strictly epigenetic. If we take one of those FK506 resistant epimutants, let it revert, and select epimutants again, they occur at the same frequency. It's 30% of the population. So we think that it's strictly epigenetic. But as I mentioned, for the pure F and pure G epimutants, those occurred in an RNAi mutant background as a genetic helping hand. And so you can envision pathways that have both a genetic and then an epigenetic component occurring as well. So from one common selective pressure, we found 2 different types of isolates: Mendelian mutants that are stable and epimutants that are RNAi dependent and transient and unstable. And we think that this allows organisms to sample a much broader phenotypic space. They don't make a permanent commitment to the loss of FKBP12. They just shut it down for the period of time they need to, and then it can be restored when the selective pressure is relaxed. Now, I want to close by generalizing this. And epimutations were discovered originally in plants. And this is a snapdragon. The wild-type snapdragon has bilateral symmetry across the flower. This is a variant that was discovered by Carl Linnaeus, the famous botanist and phylogenist, something like 4 centuries ago. It has radial symmetry. And the report of the gene just over 20 years ago showed that the pyloric flower, it's not a mutant, it's an epimutant. There's no nucleotide changes in the underlying gene. It's extensively heavily methylated, and it's stable enough to have been passed through flower crosses for centuries. So it can be stable through meiosis, and occasionally it'll revert back to wild type. Epimutants have also been described in humans. So you're probably familiar with a human genetic cancer predisposition syndrome. It's called either hereditary non-polyposis colorectal cancer, HNPCC. That's a bit of a mouthful, so it's now more commonly known as Lynch syndrome. these individuals and families have mutations in the mismatch repair pathway, and that leads them to have inherited cancer predisposition. In some of these families, though, they didn't find any mutations in the suspect genes. And you can see 3 different reports of epimutations occurring in either MLH1 or MSH2. And there are a variety of other reports of human epimutations. Again, in this case, it's not RNAi, it's methylation of the gene that shuts down expression. Very recently, there's a paper published by Robin Alshire in Nature. This was published September 9th of this year. And they identified epimutations in Schizosaccharomyces pombe by selecting for caffeine resistance and then screening for isolates where the resistance is unstable and showing that their resistance is dependent on a heterochromatin factor, CLR4, that makes the heterochromatic mark H3K9 dimethyl. When they chipped in the genome for the location of H3K9 dimethyl, you light up the centromeres of the 3 chromosomes as you expect, but in each of these unstable resistant isolates, another region of the genome lights up as having been turned in to heterochromatin. And in this case, there's 4 or 5 genes that are covered by the heterochromatic peak, and one of those genes has already been implicated in caffeine resistance. So you can have epimutations that result from RNAi, or for DNA methylation, or for heterochromatin formation. And it suggests that other organisms, other fungi, oomycetes, human pathogens, plant pathogens that have heterochromatin or RNAi might be subject to these types of pathways. So I want to summarize then, we focused on RNAi and epimutations as a form or a generator of phenotypic diversity, but there's a panoply of other factors that can also contribute to microbial diversity, including things like HSP90 as a capacitor for evolution, prions, aneuploidy, mutators and hypermutators, and sexual and parasexual reproduction. And all of these contribute to generate a palette of diversity that selection can act upon in promoting diversification and evolution of both populations and species. And there's 2 PLOS Pearls reviews that summarize other aspects of these diversity generators in case anyone is interested. I want to close by thanking the members of the group that contributed to this. So the Epimutation project started when Cecilia was a rotating student in the lab and then was continued by Sylvia as a postdoc. And Sue Chan was a critical component in working on Mucor in the lab. Zanetta did all of the animal work and the pure F and pure G epimutants. Blake Billmeyer, who's now at the Stowers Institute, was the bioinformatic muscle for the entire project. This was a long-term collaboration with Rosa and Santiago and Curo in the University of Murcia in Spain, and Carlos and Maribel have been contributing critical data analysis to the latter parts of the project as part of Victor Gar's lab at the same university. And I want to thank all the members of my group. I'd like to acknowledge the long-term support from the NIH, including a key training program that Teresa mentioned. We're very grateful for all of this support. And I'd like to also highlight that this is a program that's now supported also by the Canadian Institute for Advanced Research called Fungal Kingdom, Threats and Opportunities. I'll stop there and be happy to take any questions if there's time.
Thank you very much for a wonderful seminar. Thank you. We have a question from Eric and then from Gillian in the chat. Eric.
No, thank you very much. So a quick question. So if you look at your epigenetic resistant organism and then essentially make an siRNA library, do you think that you're going to see a depletion of lots of different siRNAs? You know, is this like a sort of a global phenomenon that you're having a control of lots of the siRNAs? Or is it something specific to the resistance of the drug that you're testing?
So from the libraries that we've made for the fkbP12 resistant mutants and for the 5FOA resistant mutants, essentially all we see is small RNAs increasing in abundance to those target genes. All the other small RNAs that we see across the genome seem to be at the same level of abundance in the wild type, the resistant isolate, and the revertant isolate. So the way we're thinking about it is if you look at a whole population of Mucor, say 1 billion cells, there'll be a cell here that has more small RNAs to one gene, and there'll be a cell here that has more small RNAs to another gene, and a cell here that has small RNAs to another gene. But we're not sure that there would be an intermediate that had, say, a lot of small RNAs across the genome. And we may be thinking about that in the wrong way, but we're thinking of it similar to how mutations occur. But it could also be the case when you're growing a culture that you get bursts of mutation as well. So for the ones that come out of the brain, we've only analyzed those CFU as a whole population. And we see 7 genes that have an increase in small RNAs. And what we don't know is, does every cell coming out of the brain have small RNAs increased against all 7 of those genes? Or by analyzing it as a pool, have we combined 7 different epimutants in our analysis? So we want to repeat that experiment and include colony purification as part of it to begin to address that.
So this sounds like more like a chromatin structure kind of change then? Would that be your thought?
We actually don't think there's going to be a change in the chromatin structure. We've been struggling with some of the reagents to be able to prove that definitively. It's certainly possible that there's some heterochromatin, but the fact that the trigger is the mature mRNA, we think means that it might be post-transcriptional and it might not involve heterochromatin, because if you were to form heterochromatin, you might imagine you wouldn't have any RNA there to continue the process.
Thanks.
Gillian, you had a question that's in the chat, I see. Is there any association or connection of the epimutation phenomenon to the whole genome duplication? That's a great question. So we don't know. And so when we tried to generalize this to other species, we spent some time trying to look for epimutations in Neurospora and in Cryptococcus unsuccessfully. And we then went back and said, let's look at the closest species we can look at. So we are thinking about broadening that now to these 16 phylogenetic species, but they've all undergone this whole genome duplication event. So we need isolates that would undergo this that are further away. And I think we're going to try harder with Cryptococcus, and we've been hoping that somebody would find this in Neurospora or Aspergillus in the meantime.
Yeah. You preempted my second question. Has this been demonstrated for any plant pathogen? But
not that I'm aware of. And Sylvia's paper was published in 2014, and I thought there might be a flurry of reports that followed that, but it took some time, and Robin Alshire's, I think, is really the first example. There is another paper that I skipped over at the end there in the interest of time, where they characterized fluconazole-resistant Cryptococcus isolates, and about half of them turn out to be aneuploid for one chromosome or another, but the other half, they're unstable, and they don't have any aneuploidy, and they don't know what the mechanism is. So we would like to make small RNA libraries of those isolates and see if they might be epimutants. And I think that could be a good starting point. I think for plant pathogens, given Robin Alshire's paper, you could entertain the idea that they might be either heterochromatin or they might be RNAi, or given the plant and the human work, they might be DNA methylation.
Right.
So any of these mechanisms could give rise to something that might be unstable at some level.
Thank you.
Any other questions? I have a question then. So Mucor and Mycotina are famous for their resistance to various antifungal agents. Do you think that there is a common mechanism related to your recent discoveries, or the ability to withstand the pressure is by different mechanisms?
So it's one of the defining features, I think, of the mucorales as a human pathogen, and they are intrinsically resistant to a lot of drugs. And when you look at RNAi mutants that couldn't form an epimutation, they don't become more sensitive to those drugs. So we think there are other reasons. In the case of some of the azoles can have a short or a medium or a long tail. And there are some amino acid substitutions in ERG11, the target of the azoles, that make Mucor resistant to some azoles, but still sensitive to things like isobuconazole. Tobias Hall was visiting one time and we presented this story to him and he said, it's pretty interesting, but it would be a lot more interesting if you could show this would be relevant to a drug we actually use in people. And so we are planning to look for epimutants that might make you resistant to amphotericin B or posaconazole or isobuconazole. And it turns out you can get loss of function mutations in the ergosterol biosynthetic pathway that could do that. So an ERG6 mutant should be amphotericin B resistant, and ERG3 mutant or epimutant would be azole resistant. Some of those one of those genes is triplicated in Mucor. So that might be a unique case where it'd be easier to get an epimutant than a loss of function mutation. So when Carlos and Maribel arrive, that's part of what we're planning to pursue. And that might then link it to mechanisms of drug resistance. There's a lot of drug resistance occurring, I think, in both plant and human fungal pathogens that's in response to azole pressure in agriculture. This is a huge problem with Aspergillus fumigatus, and there's the suspicion that it might be contributing to Candida auris drug resistance. And Mucor infections come from the environment. So we're also going to look at agricultural azoles as well.
This has been a production of Cornell University, on the web at cornell.edu.